Welcome back. This video we are going to discuss about tissue culture and spore formation. Last class we have seen vegetative propagation and artificial propagation. So in artificial propagation we have discussed about cutting, layering and grafting. So in this class we will see tissue culture. So what is tissue culture? So the name itself says tissue culture means tissues are cultured. So which tissues are cultured? Plant tissues are cultured. So if we see the definition, few plant cells, plant tissues are placed in growth medium with plant hormones. Actually a plant grows in soil. Soil gives minerals and all the properties which are used for growing. So here we are using growth medium as the plant hormones. So plant hormones, with the help of plant hormones, they are used as growth medium. Plants are grown with the cells and tissues of plants. Okay, new plants are grown. So suppose if you think a small stem is there, from that small tip of the stem or a small piece of the stem, hundreds and thousands of tissues we can take, and hundreds and thousands of cells also we can take through microscope, and those cells and those tissues are kept in growth plant hormone, and they are given some favorable conditions for growing. Afterwards the new plants will be grown and they are used economically very well. So this tissue culture is economically beneficial for the farmers. So what are the important points in tissue culture if we discuss? This tissue culture, mainly all the new plants grows in short span of time or short period. So within short period we can get thousands of new plants. So that is the use of tissue culture, and mostly scientists basing on tissue culture they are growing many number of plants. So next, coming to the next topic, it is spore formation. The spores are nothing but which are produced from the fungus. So these spores are also this reproduction is also through asexual reproduction only. So we can see these spores on rotten fruits, vegetables and bread slices. So if tomatoes or any other vegetables or even fruits also, if we keep for many days in the house, so we can see the fruit part will be cut and you can see whitish, grayish or blackish fungus formed on the rotten fruits or spoiled fruits and vegetables. So even if you take a bread slice and keep for many days in a polythene cover like that, so after a few days we can see that fungal formation on the bread. So these formation powdery substance will be there if you touch it with your fingers. So that powder, if you keep under a microscope and if you observe it, we can see rhizopus or mucus, or as a whole we can see it is nothing but the powdery substance is fungi or fungus. So this fungus is of many types, rhizopus and mucor. So in non-flowering plants also we can see this. So non-flowering plants like fern and moss plants, fern is the example and moss plant, there also we can see the formation of spores. So if we see fern plant, it is an ornamental plant. If we see fern, so the plant is like this. This is a base and these are the roots. So the root part you can see below the soil. So it is a bunched plant, ornamental or indoor plant. Many of them use as indoor plant also. So the twig, the leaves are leaflets, leaves are like this. So it is like this, the fern plant. But older stems or older leaves you can see the sporangium, and the sporangium, if you observe under microscope, that sporangium you can see many number of spores. So the leaves are like this. Okay, so one of the leaf if we see, it will be like this. So the leaves are in this shape. So the older leaves, if you observe, we can see sporangium on that. So it is light greenish in color, this plant, and most of the houses we can see this as a indoor and ornamental plants. So this also reproduces with the help of spore formation. So the leaves resemble like this, and if you see the base, the older leaves, you can see some patches or spot like structures on the leaf. So on the newly formed leaves, these are not seen, only we can see on older leaves. So the patches will be like this. These patches or the spots are nothing but the sporangium. So the whole twig of the leaves, it is called as sporophyll. It is called the sporophyll. Means all the leaves in this twig contains the sporangium. So one of the sporangium, so this rounded structure, this is sporangium. So if we emboss and see, the sporangium will be like this. So number of spores will be present in the sporangium. If we see under microscope, we can see. Once the sporangium is matured, a cut or break occurs or happens, and all the spores are thrown out through air. So the dispersal of these spores is through air. So all the spores will come out, they fall on the soil or the substratum where the fern is growing, and one sperm, the spore falls here. So again new plant arises here. So these are all nothing but spores. Okay. So this is about the fern plant. The plant is there. So it reproduces with the help of the spore formation. So the whole all the leaves where the twig, all the sporangiums are there in the leaf, that twig is called as a sporophyll. Sporangium you can see on the back side of the leaf, and one sporangium, if you emboss and see in the microscope, you will be seeing microscopic structures, nothing but the spores. So through dispersal of these spores, the formation of a new plant will be occurring. Wherever the spores fall on the substratum or on the soil, new plants will come from that. Now if we see rhizopus here, so in our class we are having a very important experiment regarding this rhizopus and bread mold. So if we see, there is a lab activity for 4 marks in this chapter. So they will find out the rhizopus, how you extract the rhizopus which is formed on bread. So for that activity, if an activity is there, we have to write the activity in the form of aim, materials required, the process how we do or the procedure, and the precautions what we take, the precautions, and final the results or our observations. So it is a 4 marks question. So how you find out rhizopus or fungus on a bread mold? So for that we have to aim, we have to write as finding rhizopus or fungus on bread mold. So what are the materials required? So here the process is, you have to take a bread mold and place the bread, slice in a polythene cover and keep aside for 2 or 3 days. Now and then you open the polythene cover and sprinkle little amount of water, so that the place is the slice is damp or wet. Then after few days, if you observe, so if this is the bread slice, you can see dark patches here and there. So small dark patches will be seen here or all over the bread. So powdery patches, fungal patches. So if we touch the powdery substance, you can see. So with the help of a forceps, you take out the powdery part and observe under microscope. Then we can see rhizopus. So that is the process, and now we will draw how the rhizopus or the rhizopus appears. So if we draw that, say actually rhizopus, it is so this is rhizopus, root system is there. So some stem like structures are there and a bulged part will be there. So this is the rhizopus, and this connection between two rhizopus, this is called as hyphae, and these are the root structures, and the bulged part nothing but sporangium, and in this sporangium also clusters of spores you can see. So these are the clusters of spores. When the sporangium is matured, one of the sporangium will have a cut and all the spores will be falling on the substratum. So these are the spores. So this is the rhizopus. So these spores, they fall on the substratum. Substratum means here the substratum means here the bread slice. So when they fall on the bread slice, again new rhizopus will be growing. So all this, so a little bit starts here at the corner of the bread slice, all over the bread it will be occupying the space. So or it will be, if a little bit you can see on one corner, by the next day it will take, it easily passes into whole bread or it covers the whole bread. So in this way the rhizopus is formed. So here what we have to write the precautions: do not touch the rhizopus or the fungus which is formed on the bread with the hands, or use only forceps for extracting the rhizopus or extracting the fungus to see or to observe under microscope. After observing under microscope, what are your observations? We can see some clusters of rhizopus which are formed on the bread slice, and you can observe sporangium, and you can see the ruptured sporangium which contains spores. So this is the activity we can see. So this is an important activity in this chapter, and mostly they may ask diagrammatic questions, means they give the diagram and they may ask to write out the parts of this diagram, or as a whole activity will come for this. If activity comes, we have to write in this form. We have to write the aim, materials required, and the process with precautions and observations. So in this class we completed this. So here by this we completed sexual reproduction. From the next class we will see sexual reproduction. Thank you.